Okay, so I think I'm set up now. Hi everyone, my name is Adrianne Watson and I'm a PhD student at the University of Alberta. So today I'm going to talk about how neural stem cells build and repair the brain. So this talk is going to be divided into three parts. So I'll start with just going over what stem cells are, their characteristics, and how they were discovered. And then I'm going to move on to talking about why we study stem cells and how they're important models and potentially treatments for different diseases. And finally, I'm going to talk about my own research, which is how we can engage neural stem cells for brain building and repair. So let's start with the question, yes or no, was Canada a pioneer in stem cell research? So the answer to this is yes, absolutely. Canada was a huge player in the establishment of stem cell research. This was primarily due to the amazing work by three Canadian scientists in the 1960s, Dr. James Till, Dr. Ernest McCulloch, and later on their graduate student, Dr. Andrew Becker. This was done at the University of Toronto. So these biologists were the first ever to demonstrate the existence of stem cells. So how did they do this? Well, they started by isolating cells from the bone marrow of one mouse and injecting them into the bone marrow of a different mouse. But 1 to 2 weeks later, they saw that the spleens of these mice grew these little nodules, which they called spleen colonies. When they looked at the spleen colonies under a microscope, they saw that the cells had differentiated into three primary blood cell types: red blood cells, white blood cells, and platelets. Later on, it was Dr. Andrew Becker that figured out these cell types were genetically identical, which showed that each colony of these diverse cell types was derived from one single stem cell. So stem cells have two very important functions. The first is self-renewal or proliferation, which is shown on the left of the picture here. So this is the ability of a cell to go through numerous cycles of cell division while maintaining their undifferentiated state. This is important to maintain a consistent pool of stem cells. The second important function is differentiation. So this is the ability of one cell to change its identity to adopt a more mature or specialized cell type. And sometimes they have to go through a series of these differentiation stages or changes until they get to their final state. So since Till and McCulloch, there have been many different stem cells that have been discovered. So there are skin stem cells, which are maintained in the epidermis and contribute to hair growth and skin repair. We have induced pluripotent stem cells, which are fibroblast cells, which are modified to adopt a pluripotent stem cell-like phenotype, and these are often used in research. We have hematopoietic stem cells, or blood-derived stem cells, and this is what was first isolated by Drs. Till and McCulloch. We have embryonic stem cells, which are capable of becoming any cell type in the body during development. Cancer stem cells, which play major roles in the development of tumors and different cancers. And finally, neural stem cells. So these are very important for brain development, but they're also required throughout life for learning and fine-tuning of cognitive circuits. Okay, so to really understand what stem cells are, we have to first understand how humans and other mammals develop. So first, a sperm cell will fertilize an egg or oocyte cell and create a zygote. The zygote then undergoes mitosis, which is where it replicates its chromosomes, which carry the two parental DNA, and divide into two identical cells. This process continues, and once it reaches the 16-cell stage, we call it a morula. So the cells in this stage are totipotent, which means that these cells are capable of making the organism and the placenta. So these cell divisions continue, and after about 5 to 6 days of cell division, we are left with a blastocyst. So the blastocyst has an outer cell layer, which is shown in yellow on this diagram, which is called the trophoblast. So the trophoblast goes on to make the placenta. Inside the trophoblast is a group of cells called the inner cell mass, and these are shown in blue. So these cells are pluripotent, which means they can make any cell type in the body, but they can't make the placenta. So any organism between the zygote and the human fetus stage is called an embryo. So we call these inner mass cells embryonic stem cells. So at this point, the embryonic stem cells then further differentiate into more specialized types of stem cells that make up the different organ systems of the body, such as the circulatory system, nervous system, or immune system. So today I'm going to zoom in on the nervous system. So the embryonic stem cells that make up the nervous system come from a part of the embryo called the ectoderm. So as the fetus develops, the ectodermal stem cells become more and more specialized or mature, and then they eventually become adult neural stem cells. These adult stem cells then mature even further to adult progenitor cells, where they sit in specific areas of the brain called neural stem cell niches. And these niches are maintained from birth throughout the entire lifespan. So adult stem cells are mostly what we call senescent, or not actively renewing or differentiating. However, in certain circumstances, like when we learn something new or our brain is injured, these stem cells are recruited to self-renew and differentiate into one of the three cell types that populate the brain. And these are called neurons, oligodendrocytes, and astrocytes. So a little bit more about each of these three cell types. So first are neurons. So these are the cells that receive information from the environment, process it, and transmit information to our body. Oligodendrocytes are the cells that are responsible for the production of myelin or white matter. So they sit between the neurons and kind of secrete this protective layer over top of the axons of neurons. And finally, we have astrocytes. So astrocytes are cells that also sit between the neurons and help support them in day-to-day activities. So why is it important to study brain development? So it's important to study brain development so we can understand how neural circuits that are important for cognition are generated. So this will help us understand what kind of goes awry in neurodevelopmental disorders such as autism or schizophrenia. It might also give us clues as to interventions that we can do to help restore healthy brain development in individuals with these disorders. Finally, if we understand how the brain is put together, we'll have a better idea of how to help repair the brain after damage due to either injury or disease. So my project is primarily focused on this last point. So my work is focused on a neurodegenerative disorder called multiple sclerosis, or MS. So it's characterized by these different plaques or spots in the brain where white matter is damaged or degenerated. So it's associated with a number of different physical and psychological symptoms such as brain fog, fatigue, and mobility issues. And for reasons that we don't fully understand, Canada has one of the most high rates of MS worldwide. So we don't know exactly how MS is caused. Most of the evidence points to a dysregulation of the immune system, such that it causes damage to the central nervous system. So in this analogy, you can think of the neurons as these wires transmitting information, myelin as the insulation that helps the information travel faster and also protects the wires, and oligodendrocytes are like the myelin builders. So what happens in multiple sclerosis is the immune system first attacks the myelin and breaks it down. This causes neurons to be uncovered. The immune system also targets oligodendrocytes, so these cells are no longer there to help repair the myelin that's been lost or damaged. And finally, the immune system will attack and kill the neurons directly. So we think it's a combination of all three of these events that leads to the neurological symptoms that we see in multiple sclerosis. So our lab thinks that if we can somehow encourage these neural stem cells to become oligodendrocytes, we could then repair brain damage in diseases like multiple sclerosis. So how do we do this? How do we encourage these neural stem cells to become oligodendrocytes? So as neural stem and precursor cells kind of trundle on throughout their lifespan, they eventually reach this point, this fork in the road, where they have to choose whether to become an astrocyte, an oligodendrocyte, or a neuron. But how do they know which path to choose? So there are a number of different factors that regulate neural stem cell fate and function. These are largely signals found in their microenvironment. So this includes signals from the extracellular matrix, so the outer layer of the cell, signaling molecules. So this can include neurotransmitters like serotonin or dopamine, cytokines, so these are your immune molecules or inflammatory molecules, growth factors, and other cell populations. So I'll be talking mostly about regulation by other cell populations for my project talk. So early on in development, we have a lot of these signals that tell stem cells to either self-renew or differentiate, and this is what builds the brain. However, later on in life, most of these signals are telling our stem cells to undergo senescence. So this quiet, very non-dividing, non-differentiating state. And especially in neurodegenerative diseases like multiple sclerosis, we have a lot of these signals that are interfering with the ability of stem cells to differentiate and repair the brain. So our lab is trying to figure out a way that we can wake up these neural stem cells and encourage them to differentiate into oligodendrocytes. So my supervisor identified a special type of neurons called interneurons that secrete a molecule called Fractalkine. So we learned that it is present in healthy brains, but there's far less of it in brains that are afflicted with multiple sclerosis. We also found that neural stem cells express the receptor for Fractalkine. So there's potential for Fractalkine to act on neural stem cells and cause some sort of effect. So my project is looking to see if Fractalkine can engage these neural stem cells, wake them up, and encourage them to become oligodendrocytes. So I did this with 3 different methods. So with anything new in science, we have to start from very simple systems and move to more complex. So the first thing I did was I just added Fractalkine to neural stem cells that I grew on a plate. Then I added Fractalkine to brain slices that I grew in a dish. And then I actually treated healthy mice with Fractalkine. So the next few slides, I'm going to go through what I found in each of these systems. So first, looking at neural stem cells. So to do this, I isolated neural stem cells from the brains of mice and grew them in a plate that looks like this. So each well has about 75 to 100,000 neural stem cells. And then I add about 1/2 ml of this vitamin juice that helps the cells grow as if they were still in the brain. So then I added Fractalkine, and I assessed if Fractalkine was helping the cells proliferate, differentiate, if it was telling them to die, or was telling them to undergo senescence. So here in the pictures, I marked the cells with different colors. So the gray blobs just represent any cell, whereas the green spidery looking ones are oligodendrocytes. So what I found was that when I added Fractalkine to these neural stem cells, it increased the number of oligodendrocytes they were able to form. So then I moved on to my brain slices. So this is a very cool system where we can take little sections of a mouse brain and add it to our nutrient juice. And then we add Fractalkine and we can assess how the neural stem cells behave in a living tissue. So this is a picture of what that looks like. Here I've marked myelin in green and all the little blue dots are cells. So similarly here, when I added Fractalkine to these brain slices, I saw there was an increase in the amount of myelin that was formed. So the last thing I did was treat healthy mice with Fractalkine. So to do this, we surgically install a mini pump that infuses Fractalkine directly into the brain. A little capsule sits on the back of the mouse that contains the Fractalkine, and then it's continually infused through a pump directly into the brain over the course of about a week. So then what you're looking at in the pictures is a very thin slice of a brain that we've added different colors to visualize the different cells. So oligodendrocytes are in red, and every newborn cell is in green. So every cell that is newly formed from the time we started treatment, the time we finished treatment is in green. And so the newborn oligodendrocytes are these yellow cells here. So when we gave the mouse Fractalkine, we saw a significant increase in the number of newborn oligodendrocytes that were formed. So all of this shows that Fractalkine can engage neural stem cells in the adult brain and encourage them to produce more oligodendrocytes. And this causes an increased amount of myelin in the brain. So if this also holds true in brains afflicted by multiple sclerosis, this could repair the underlying damage that causes multiple sclerosis symptoms. So this is just one of the few ways that we can use neural stem cells as a way to help repair or correct brain development and brain damage. So this is a picture of my lab and everybody that helped generate this data. And all the names that you see in bold are people that actually contribute to the data that I showed today. So my supervisor, Dr. Voronova, Tim Foots, our lab technician, the postdoc in our lab, Dr. Amida, and the undergraduate student in our lab, Jessica Lee. And at the top are all the funding agencies that help support this work. So thank you guys all so much for listening to my talk. I know I won't be able to answer questions in person, but if you have any questions about my work or about the presentation, feel free to email me at the bottom here. Awesome, thanks guys.